Okay, are there any questions? Can somebody confirm that you can hear me, please?
Yes.
Thank you. Let me turn my volume up. Let me share my screen. All right, you should be able to see my screen now. Well, that's kind of big. So today is May 18th, and we should be finishing chapter 8 today, and hopefully starting a little bit of chapter 14. Any questions about any of that? There is no lab today. You should be working on your unknown project. I will log in for the first 15 minutes of the lab just to answer questions. But after 6:45, I'll log out. So if you have any questions, make sure you log in the lab between 6:30 and 6:45. All right, any questions about any of that? Any questions about what we're going to do? If not, I'll begin chapter 8. All right, last time we were talking about the operon model of gene expression. Is that correct? I'm not hearing anything, but I'm pretty sure that's where we were. And I did mention that an operon is sort of like a super gene. It does include the structural genes, which is normally what we think of when we talk about a gene. And then the operon control has the controlling regions around that structural gene, such as the P and the O. The P stands for a promoter. We've actually already talked about that. And the O stands for the operator. We will talk about that. It's not shown on here, but the terminator, which will follow the structural genes, is a part of the operon. And this is the lac operon I'm showing you, which in prokaryotes has 3 structural genes in the lac operon. It has the Z gene, which is the gene that encodes for the enzyme lactase. It has the Y gene, which is the gene responsible for making the transport protein that brings the sugar lactose into the cell. And then the A gene is another gene involved in the metabolism of the sugar lactose. And the A gene, you don't need to know this, but the A gene actually helps convert beta galactose into the sugar glucose. Any question about any of that? I probably should make you know that, but I'm not going to. And that is, I've mentioned that glucose is the base sugar, and usually all other sugars are converted into glucose and then sent down aerobic respiration in the normal way. And I've mentioned that most carbohydrates are converted into glucose and then sent down the aerobic respiration in the normal way. And so if one sugar is metabolized, like in the triple sugar iron test, you know that glucose is also metabolized, because if the organism can use one sugar, it can use glucose, because glucose is the base sugar. Any question about any of that? And that's just something you should know for the lab, that glucose is the base sugar. All right, let's talk a little bit more about the regulation of gene expression. The lac operon is an inducible operon. There are two types of operons. There are inducible or induction and repression. We're not going to talk about repressed repression of the operon, but for the lac operon, the lactose genes or the lactase gene are normally turned off, and they're only turned on when the cell needs the gene to be turned on, when it needs the expression of these genes. And that's why we call it an inducible operon. A repression is where the gene is normally turned on, but it can be turned off, and you use repression to turn it off. Any question about that? All right. Normally, the lac operon is turned off. And so when the polymerase binds to the promoter, and that's what the P region of the operon stands for, it will then move down the gene, and it's turned off by a regulatory gene, which is called I. And I don't remember if I mentioned it earlier. We have I shown here. Usually the regulatory gene, which is I, is not near the gene that it regulates. I is not a part of the operator. It is its own operon, and it has its own promoter and probably operator. And obviously I is the structural gene and has its own terminator. And this gene is regulated separately of the lac operon. Any questions about any of that? As I stated, the I gene is normally not close to the gene it regulates. In the case of a eukaryote, the regulator is usually on a different chromosome than the gene that it's regulating, meaning the operon it's regulating. In the case of the lac operon, the regulatory gene happens to be very close and is really right next door to the gene that it regulates. And that's not commonly seen. Any questions about any of that? All right, so when the I gene is expressed, meaning it's turned on, it makes its gene copy, it's transcribed, messenger RNA is translated into a protein. This protein is a repressor protein, and it is active. And that repressor protein binds to the operator of the lac operon. Now, when the polymerase binds to the promoter and then moves down the DNA, the movement of the polymerase is blocked by the repressor protein. And so the polymerase doesn't move any further than the repressor protein. And that shuts down the lac operon, meaning the Z, Y, and A gene will not be expressed because the polymerase cannot reach it. Any questions about any of that? All right, here's a question for you. Why would E. coli, as well as most other bacteria, normally want to shut off the genes responsible for the sugar metabolism of lactose? Anyone have a guess? Well, I'll give you a hint. Where do you find the sugar lactose? Come on, everyone should know that one.
In the intestine.
Well, not necessarily. If someone's on a special diet and they don't consume the sugar lactose, they're not going to have the sugar lactose in their intestine. My question is, where do we find the sugar lactose? Meaning, what food products do we find the sugar lactose in?
Dairy?
Yeah, dairy products. I am lactose intolerant, so generally I don't eat much lactose. So in my gut, I don't often have the sugar lactose in there. And that's just my point, and that is in my gut, you're not going to find the sugar lactose very often because I don't eat lactose. The only time I do is maybe a little bit of yogurt and a little bit of cheese. I don't ever touch milk because I'm lactose intolerant. How often would E. coli find a dairy product? So we're talking about a bacteria. If it's growing on an apple tree or it's growing in the soil, are those E. coli going to see the sugar lactose? Come on, people, that's a yes or no question.
No.
Yeah, they're not going to see it unless the E. coli happens to be on a dairy farm, or if somebody eats dairy products and the E. coli is in your gut, then the E. coli would see the sugar lactose. Okay. But most of the time, E. coli, if it's on an apple tree or in the soil or most of the places it is in the world, other than in a gut of someone, you're not going to see dairy products. And even if you're in the gut of a pig, for example, I don't think pigs often eat dairy products. I could be wrong, but I don't think they often eat dairy products. So most of the time, the E. coli is not going to see the sugar lactose. And so why would it make the genes, the enzymes for metabolizing lactose if E. coli is not going to see it? And that's why E. coli normally shuts down the lac operon. Any question about any of that? All right, let's move on. Well, what happens when the E. coli does see the sugar lactose? You need some mechanism to turn on the lac operon then. And here it's how it is done. The repressor protein gets made, and then when the sugar lactose is around, the sugar lactose is an isomer of the sugar allolactose. And if you don't remember what an isomer is, it's the same chemical structure. It's just one is left-handed, the other is right-handed. Is that clear? And whenever the sugar lactose is around, the sugar allolactose is around. Allolactose will bind to the repressor protein and inactivate it. If the repressor protein is inactivated, it cannot bind to the operator. And then when the polymerase binds to the promoter, it'll move down the DNA and transcribe the Z gene. And that's how the lac operon gets expressed. Any questions about any of that? Now, there's something unusual about this messenger RNA. And as you can see right here, we get one transcript product, one messenger RNA from this operon. But how many proteins do we get from that messenger RNA? Come on, people. It's showing right here. 1, 2,
Three.
3. Thank you. This one here, black beta galactosidase, is another name for lactase. Permease is the transport protein that brings the sugar lactose across the cell membrane. And then acetylase is the enzyme that helps convert beta galactose to the sugar glucose. Okay. The point is, in the lac operon of bacteria, you get 3 proteins made, and that's because there's 3 structural genes in the lac operon in prokaryotes. Now, in a eukaryote, we do have all 3 of these genes: the lactase enzyme, the transport protein to bring it into the cell, and then the transacetylase to help convert the sugar beta galactose to the sugar glucose. So we have all 3 of these structural genes. It's just in eukaryotes, there's only 1 structural gene in an operon. So in eukaryotes, we have 3 operons instead of 1, which would be the lac operon in prokaryotes. In eukaryotes, I don't even know what they call them. One might be the lac operon, the other the permease operon, the other the transacetylase. I don't know what they call them, but that would be my guess, is what they call them. All right, here's another question for you. If you remember back here, we had the repressor protein bound to the operator. How does that come off? Does the inactive repressor protein bind to the repressor protein bound to the operator? I'm going to tell you, no, it does not. So how does that inactivate a gene that's currently already turned off because the repressor protein is bound? How does that change from the gene being turned off to the gene being turned on? This is actually a more difficult question. So if you don't know the answer, that's all right. What happens is that the binding of this protein is not absolute. It actually is a stoichiomic reaction. And every once in a while, this protein falls off the operator. If there's more repressor protein around, it'll bind to the operator and then shut down the lac operon. However, if the sugar lactose is around, then the sugar allolactose is around, because they're the same sugar, really. And it will bind to the repressor protein that falls off the operator, inactivating it. And now there's no repressor protein to bind to the operator. And so a polymerase that binds to the promoter can then move down and express the gene. Any question about any of that? All right. Another point here is, what happens to this RNA polymerase that gets blocked by the repressor protein? Is it permanently blocked until that repressor protein falls off? Anyone have a guess? That's a yes or no.
Yes.
No, actually, it does not wait for the repressor protein to fall off. The RNA polymerase is also binding to the DNA by a stoichiomic reaction. So every once in a while, the RNA polymerase will fall off, and then it will just bind to another promoter and try to transcribe that gene. And it could bind to the same promoter and be blocked again. But my point is that both the polymerase and the repressor protein fall off of the DNA every once in a while. And so that is an essential part for the regulation of the lac operon. Because otherwise you'd tie up all the RNA polymerase. They'd be bound to a promoter and then they're stuck. Then you don't have enough polymerase around. Let me see if there's anything else I wanted to discuss. I talked about that. Nope, let's go ahead and watch a little video on the lac operon. Well, there it goes, just a little slow.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling.
All right, that's not exactly correct, and that is the repressor protein does not bind to the the sugar allolactose does not bind to the repressor protein when it is bound to the operator. That's shown in the video, and that's not quite correct, but everything else in the video is correct.
Region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any questions about that little video? Can someone answer? Did we see that earlier, this video, when I was talking about translation? Or is this a separate video?
Yeah, we saw it last lecture.
Okay. Like I said, I thought that video that I showed you for protein translation was the wrong video. And I don't know why it went to this, because initially it said closed or a video isn't there. And then it went to this one. And it did sort of explain a little bit about translation, but I'll have to straighten that out. Let me make a note of that so I remember to do that. Sorry about that, but all right. Let's close that down. Any questions about the lac operon or the video? If not, let's move on to the topic of mutation. A mutation is a change in the genetic material. Mutations may be neutral to the organism, they may be beneficial, or they may be harmful. Most mutations are either harmful or neutral, and we'll explain how they can be neutral. A mutagen is an agent that increases the rate that mutations occur. So it increases the mutation rate. And there's different agents, like a chemical agent, that can be a mutagen. Mutation does happen in the absence of all other factors, and we call that a spontaneous mutation. It occurs in the absence of a mutagen. And spontaneous mutations happen all the time. It's just a mistake of the DNA polymerase putting in the wrong nucleotide. There are different types of mutations. A base substitution, also called a point mutation, is where one base is inserted that is incorrect, meaning you're putting in one nucleotide base incorrectly. This can be a silent mutation, a missense mutation, and a nonsense mutation, and we'll talk a little bit about those in the next slide. Anyways, the base mutation is where only one base is changed. A frame shift mutation is where one or more bases are added or are subtracted. And this causes a reading change in the nucleotides. And we'll talk a little bit more about that in the next couple of slides. I'll show you an example. A macromutation is a topic that is not covered in your book, and I do not know why they do not discuss it. But a macromutation is different than a base substitution or a frame shift mutation. A macromutation is where an entire region of a chromosome is either added or deleted. Does anyone know of a macromutation where there's an entire chromosome that is extra? Nobody knows? All right. Like I said, that's not in the book, so be careful and make sure you go over a macromutation in my notes. This is an example of a child that they were studying to see if he was a mutant, because as you can see, let me blow that up a little bit. Spoons are sticking to his skin. And they were wondering, is he a mutant like Magneto, that metallic objects stick to his skin? And they were actually studying him. I read this several years ago, many years ago, but I never got a follow-up to it. So I don't know what the study ever found. All right, a macromutation that is with an entire extra chromosome is Down syndrome patients. They have an extra chromosome 21. And that would be a macromutation. You can also have just a portion of the chromosome which is either present or absent. And that would be a macromutation also. All right, so let's talk about a base substitution. This is where a nucleotide gets replaced. Let me see if I can blow this up. So normally we have TGT. So we normally see A, C, A, like over here. But here the DNA polymerase put in the wrong base. And this actually could be repaired. We're not going to talk about DNA repair, but the DNA can notice that this is not pairing. And so it could actually replace the newly added base. And the cell has a way of knowing which strand of DNA is the new one. And usually what it does, it cuts the new strand where it's not pairing correctly and then replaces it. We're not going to talk further about that. But the point is, if this goes through DNA replication before it's repaired, that T will then put in an A. And now we have this mutation fixed because you can't repair this because you just can't, okay? That's not entirely true either, but we're not going to talk about a base mutation, a backwards mutation. But essentially, when the DNA gets replicated, this mutation is now fixed because it's an AT pair and it should be a GC pair. And this would cause a change in the DNA. And so this mutation would be expressed. Okay, normally we'd have cysteine and then cysteine, and instead we have cysteine and tyrosine. Anyways, that's a base mutation. Any question about a base mutation? A silent mutation is where we change one base of DNA, but it does not result in an amino acid change in the protein. So this mutation has no effect on protein function. And the reason for that is because the redundancy of the genetic code, that phenylalanine, if it's UUU and it gets mutated to UUC, it still codes for the amino acid phenylalanine. This we call a silent mutation, where the DNA is changed, but the amino acid is not changed in the protein. This mutation has no effect on the host. And so it would be a neutral mutation. Any question about any of that? Doesn't give the host an advantage, doesn't give a disadvantage. A missense mutation is a change in one DNA base, and it does result in a change in the amino acid. Let me blow this up a little bit, just a little. So we have CCG. Is that right? Maybe it's GGC. And it gets converted to AGC. So this nucleotide gets changed. Instead of coding for the amino acid glycine, GGC, it now is AGC. That changes the amino acid. It's now serine. And then this protein will differ because it has a different amino acid. This is an example of a missense mutation. Any question about that? An example of a missense mutation is most sickle cell anemia, where you change one nucleotide in the DNA, and it does result in an amino acid change. I think it's in the 6th amino acid of the protein. And then that results in sickle cell anemia, assuming the patient has 2 of those alleles. Another example of a missense mutation was 10,000 years ago, the human race only had brown eyes. And 10,000 years ago, in Northern Europe, we had a missense mutation, and the blue-eyed allele came about. And now humans have the potential to have two eye colors, either bluish eyes or brownish eyes, and all the others eye colors are a derivative of bluish or brownish, and we call that the major gene for eye color. There are many other genes that give the different shades of blue. Okay, so a green eye is blue, but it has a different alleles which actually change that blue to green. But the main one, the lowercase b, is resulting in bluish eyes. Any question about any of that? Now, 10,000 years ago, it appears that the blue-eyed allele gave the individual a selective advantage. And it's possible that the blue-eyed allele was just happened to be next to another gene that had a selective advantage. And that's why the blue-eyed allele expanded in the population. And now in Northern Europe, the blue-eyed allele is actually the major allele. It's more common than the brown-eyed allele. Any question about any of that? However, today, when we look at the blue-eyed allele compared to the brown-eyed allele, the blue-eyed allele no longer has a selective advantage. So there's no selective advantage for the brown-eyed allele over the blue-eyed or the blue-eyed over the brown-eyed. But at 10,000 years ago, it appears that the blue-eyed allele either gave a selective advantage or else it was next to a gene that gave a selective advantage and it got carried because it was really close to that gene that was being selected for. It might be that 10,000 years ago, because of the change in the culture and the people and all that the advantage has now gone away. But like I said, 10,000 years ago, it appears that the blue-eyed allele had a selective advantage in Northern Europe. And today, that's not the case. Any question about any of that? All right. A nonsense mutation is a change in a nucleotide in the DNA, which results in a stop codon in the messenger RNA, so that it's a stop codon in the protein. And what that does, let's see right here, we have it's this one. We have AAG, and it gets changed to UAG, which is a stop codon. And now the protein gets made up above this stop codon. But after this, the protein is truncated because of the stop codon. This often makes the protein non-functional. It really depends on where the function of the protein is. But if you put the stop codon in the protein, you really change a lot of the protein. An example of a stop codon is some muscular dystrophy patients and some cystic fibrosis patients have a mutation creating a stop codon. And so their protein, when it's translated, it will be a truncated protein. All right, any questions about the silent mutation, the missense mutation, or the nonsense mutation? All of them are a point mutation where one nucleotide in the DNA gets changed. And there may or may not be a change in the protein. There is not a change in the silent mutation. There is a change in the missense and in the nonsense mutations. Any question about any of that? All right, let's talk about the frame shift mutation. A frame shift is an insertion or a deletion of one or more bases. So here we have AUG, AAG, UUU, GGC, encoding the amino acids methionine, lysine, phenylalanine, and glycine. And we have a frame shift mutation where one nucleotide is removed. And it looks like we're taking out the AAA and we're removing one of the A's. And now this codon will not be AAA. It'll be AAC, which will be in the messenger RNA, UUG. Everyone see that? And that G was the start of the next codon. So it takes the next base to finish this codon, and then these two codons aren't enough. Not two codons, sorry, these two nucleotides are not enough. And so it takes the next nucleotide, which is a U right here. And that means that from this frame shift mutation, all other codons are altered. So after the frame shift mutation, you permanently change the protein. And so instead of being methionine, lysine, phenylalanine, glycine, and stop, it goes methionine, lysine, leucine, alanine, and then will continue until the stop codon is reached. And that's a frame shift mutation that occurs when there's one addition or one deletion, or when there's multiple additions or deletions, with one exception. Okay, everyone understand the frame shift mutation? Where the frame shift happens, the entire protein afterwards is changed, the reading of the codons, because the codons change. There is one exception, and that is if you remove 3 nucleotides or a multiple of 3, or you add 3 nucleotides or a multiple of 3, you do not get a frame shift mutation. For example, if we were to take out UUU, then we'd have methionine, lysine, glycine, and then stop. There is no frame shift. We have this one amino acid pulled out, and so that would be kind of a missense mutation, not a frame shift mutation. Everyone see that? A multiple of 3 is not a frame shift mutation because the protein is not changed after the frame shift or after the insertion of 3 or the deletion of 3. Any question about that? Some Tay-Sachs patients are an example of a frame shift mutation where they have either an insertion or a deletion of a nucleotide. All right, let's talk a little bit more about mutations. Mutations can be caused by 2 different ways. There can be a spontaneous mutation where the DNA polymerase just makes a mistake, inserts the wrong nucleotide base, and then there can be the cause of mutation can be a mutagen, where either radiation or a chemical mutagen increases the rate of the mutation. So these are the 2 causes, spontaneous mutation, and then a mutagen causes the mutation. Let me state that I'm surprised I don't have that here. Maybe I have it someplace else. The spontaneous mutation rate is about 1 base in a billion, or 10 to the 9th bases, will be inserted incorrectly. So the spontaneous mutation rate is about 1 and 10 to the 9th bases. It's not very high, but you got to realize we have a lot of bases in DNA. And then when you're talking about the human race, our population is close to 8 billion, right? And if the mutation rate is 1 in 10 to the 9th basis, and you're looking at all the children born in a year, which may be a little less than 1 billion, but it's pretty close to 1 billion. So you know that in the human race, there will be at least one mutation. Any question about any of that? By spontaneous mutation. And that's not looking at how many bases there are in the human population. We're just looking at the number of people. So spontaneous mutation does occur, as well as mutagens causing mutation. Let's talk a little bit about radiation causing mutation. There's 2 types of radiation that can mutate DNA. There's ionizing radiation. Some examples of that are X-rays and gamma rays. These forms of radiation have a lot of energy. And when they hit DNA or a molecule that would then bump into DNA, they will cause the DNA to lose an electron, and that'll cause an ion. And if you hit a molecule other than DNA, like sodium chloride, you can cause an ion, and then that ion can bump into DNA and steal an electron from the DNA. But the point is, ionizing radiation, when it hits a molecule such as DNA, it gives that electron enough energy for that electron to leave the DNA. And then that will mutate the DNA. We can get non-ionizing radiation to also mutate to the DNA. And the only non-ionizing mutagen that mutates DNA is UV light. UV light, when it strikes DNA, let me see if I can blow this up. It causes 2 thymines that are next to each other to pair with each other. And we call that a thymine dimer, where there's 2, it's only showing 1 here, but there's actually 2 covalent bonds that occur between the thymine. And those thymines have a covalent bond between them. They do not hydrogen bond with the A that they should hydrogen bond to. That causes a problem in the DNA. This is a bubble here. And when the DNA tries to transcribe this region of the DNA, it can't. It gets stuck. The polymerase gets stuck right here, as well as DNA replication cannot replicate past the thymine dimer. So DNA replication stops right there. And that causes mutations in the DNA. We're not going to talk about the repair of the DNA, but the cell does have a way of repairing this. All right, any question about how radiation mutates the DNA? You can also have a chemical mutagen, such as nitrous acid, which mutates the DNA. Nitrous acid, when it gets next to DNA, it chemically alters a base. such as the A here. And this A is chemically still an A, but it is slightly different. And this A that's now altered does not bind with the T, what a normal A hydrogen bonds to. This altered A will now bind with a C. So the polymerase, when it's reading this, will read that A as a G and insert a C there. And then when this gets replicated, that fixes this mutation. Okay? And that is how a chemical mutagen, such as nitrous acid, can mutate the DNA. It alters this A, so it no longer binds as A, and it's actually read by the polymerase as a G, and then it puts the wrong base there. Any question about any of that? All right, here I've got the spontaneous mutation rate. I've already told you. Spontaneous mutation happens for every base out of 10 to the 9th replicated bases. Now, as 1,000 genes, excuse me, 1,000 bases is about the average gene, that means that the mutation rate per gene is about 1 gene will be mutated in 10 to the 6 replicated genes. And obviously in the human population, we have much more than 1 million people. And that tells you that there will be, for whatever gene we look at, like, I don't know, lactase, there will be a mutant in every 10 to the 6 babies. And that would be something like 1 baby in a large city. I think, if I'm correct, I believe the large cities in the United States are over 1 million. Somebody can correct me there. I don't know the population of Seattle, for example, but I believe the large cities in the United States, it's close to 1 million. And if I'm wrong, sorry, then it's close to the population of the United States, but it's something like that. Mutagens increase the mutation rate by 10 to 1,000-fold. And that means instead of 1 gene being mutated in 1 million genes, it'll be 1 gene in 10 to the 3, or 1 in 1,000, or 1 gene in 10 to the 5th. So that'd be 1 in 100,000 genes that'd be mutated. Any question about any of that? All right, let's talk about identifying mutants experimentally. You can test for a mutant with a positive direct selection. It detects the mutants because they grow or appear differently. For example, if someone like this were to go walking down the street, you'd say, yep, that's a mutant. Negative or indirect selection detects mutant cells because they do not grow. And I'll show you an example of negative or indirect selection. I've actually done this when I was in college. You can make a replica plate of let's blow that up, of a plate that has isolated colonies on it. Where you get a velvet stamper and you press the velvet onto the plate. Now the cells get on the velvet in the same pattern they were on the plate, and then you can press the velvet onto 2 other plates, and then the cells will be transferred from the velvet to the plate, and then you incubate them, and you'll get colonies growing on the plate, and they'll be the same orientation as the colonies in this plate. Well, when you stamp the plates, you can stamp 1 plate with a medium containing the amino acid histidine, and another plate with a media lacking the amino acid histidine. And then you can look to see if colonies grow on this plate that are absent on this plate. And this colony that's missing here is a mutant, and this colony requires the presence of the amino acid histidine, or it's not going to grow. So this colony here is a mutant, and it lacks the ability to make the amino acid histidine. These colonies are not mutants, and they can make the amino acid histidine. Anyways, the point is, this is negative selection for a mutant. And here we have a mutant colony. Any question about any of that? I'm trying to remember what I was looking for, and I just don't remember what mutation I was looking for when I did it. It was a long time ago. It's been a long time since I've been in college, but I was doing this when I was in college. All right, let's talk a little bit about genetic recombination. Recombination is the exchange of genes between two DNA molecules. Such as when these chromosomes get together and then exchange parts. And what happens is, this is the mother's chromosome, the blue one. This is the father's chromosome, the white and gray one. And they can come together and cross over where they actually physically cross. And then they break right here. And now the mother's chromosome right there has lost a little bit of the chromosome, the H and the I, and they gain the father's H and the I. And this is physically called crossing over, where the chromosomes come together in meiosis, and it's always the same chromosome. So this would be the mother's chromosome number 1 and the father's chromosome number 1. And they exchange, and then they exchange pieces. So in crossing over, we have the father's chromosome, capital E, capital F, capital G, and then we have the mother's chromosome, lowercase h, lowercase I. This makes new gene combinations where the capital G is now sorting with the lowercase h. And so that's a new gene combination that we didn't see before. And that just sort of shuffles the DNA, making new genetic combinations. All right, so how are recombination and crossing over, how do they differ? They're very similar, but they're not exactly the same. Anyone? Recombination is a little more general, and that is it's an exchange of genes between DNA molecules, two DNA molecules. Crossing over is physically where the chromosomes come together, cross over, and then break, and then exchange pieces. And if you don't know, that happens all the time in meiosis. So crossing over is always recombination, but recombination is a little more general. And recombination is not always crossing over. For example, suppose we had the father is Gene, lowercase g, leave the father's chromosome and go to the mother's chromosome. And now the mother's chromosome has the capital G, the lowercase g. That would be a case of recombination. It is not a case of crossing over. Any question about any of that? All right, let's talk a little bit about genetic transfer. There's vertical gene transfer, and that occurs during reproduction, where between generations of cells, the genes from the parent cell are transferred to the genes in the offspring cell. Or you could say the genes from the parents are transferred to the genes in the offspring. We're talking about humans. Horizontal gene transfer is the transfer of genes between cells of the same generation, meaning DNA leaves the donor cell and then enters the recipient cell. There are 3 types of horizontal gene transfer. There's transformation, conjugation, and transduction. Let's talk about transformation. Here we have a cell, and it's not here. The DNA leaves that cell. Usually that happens when that cell dies. The DNA is now naked DNA, and it can come to a recipient cell, and then that DNA can be brought inside the cell. The DNA can then exchange and become a part of the chromosome, where this lowercase a gene now is incorporated by this cell that never had a lowercase a gene, and then is expressed by this cell. This would happen something like if the lowercase a gene encoded for bacterial resistance to an antibiotic, like penicillin G, this cell then would pick up penicillin G resistance. So this is important. This cell is expressing a new phenotype. So we can say that this cell is transformed. It's showing transformation. Transformation is both the process of taking in DNA and then expressing it, as well as you can say this cell is transformed because it's changing its phenotype. I'm not going to talk much about the cell changing its phenotype being called transformation, but it is something that geneticists use. They do use the word transformation in 2 different ways. We're only going to talk about it, transformation, where this cell takes up foreign DNA and then gets a new gene. And here we're seeing transformation where this cell takes up a piece of DNA, gets some indigestion, and then its phenotype is changed because it expressed that DNA. Any question about that? All right. The second way that cells can have horizontal gene transfer is by conjugation. Conjugation happens in 2 different ways in gram-negative cells and gram-positive cells. In gram-negative cells is the main way we'll talk about. It happens between a F+ cell that has hyphae and a sex pilus and an F- cell that does not. The F positive cell also has an F plasmid. The F plasmid encodes for the sex pilus. And then the sex pilus literally skewers this cell and then allows the F plasmid to move from this cell into that cell. Okay. Anyways, that's how it happens, sexual conjugation in gram-negative cells. In gram-positive cells, 2 gram-positive cells actually have to touch. And I don't know what happens to the cell wall, but the cell wall disappears right there. And then the cell membrane comes together and then fuses together. And then we have a channel here connecting this cell to that cell, where the cytoplasm of this cell is connected to the cytoplasm of that cell. And then DNA from this cell can move into that cell or vice versa. And usually it's a plasmid that moves from this cell into that cell, but it doesn't have to be. It just as DNA moves from this cell to that cell. The point is, we call this a mating bridge, and that's the way that gram-positive cells can go through sexual conjugation. Let's talk about it in a little more detail. In the gram-negative cells, we have the F plus cell. It has an F plasmid. The F plasmid codes for the sex pilus. The sex pilus comes out of this cell and then will literally look for an F minus cell and then skewer the cell. That sex pilus can then act as a winch and bring these two cells closer together. They do not have to come this close together because the sex pilus is hollow and the plasmid can actually move through the sex pilus. But this is showing you where the sex pilus has brought the two cells very close together. And then the F plasmid then replicates, and one copy of the F plasmid goes into the F minus cell. The F minus cell now has the F plasmid in it, and the F plasmid codes for the production of a sex pilus so that this cell will change from F minus to F plus, and it will grow a sex pilus. And then this cell can then go on and find an F minus cell and skewer it and go through sexual conjugation. Any question about any of that? All right. Transduction is the third way that horizontal gene transfer can happen in bacteria. Transduction requires the use of a virus. And we haven't talked about viruses yet, so let me give you an introduction. When a virus infects a bacteria cell, it will inject its DNA into the bacteria. That DNA then wants to run the show in this cell, and an easy way of running the show is to degrade the host chromosome. Why? Because if you degrade the host chromosome, come on, mouse, then this chromosome is not going to run the show in the cell. What's going to be running this show is the bacterial virus DNA. And then this DNA will replicate, it'll be transcribed, viral protein will be made, the replicated DNA will go into the viral protein, making the new viruses. And then the last thing the viral DNA does is it encodes for the lysis of this cell. And when this cell bursts, it will release the newly replicated viruses, and they will go on and attach to another bacteria cell, doing the same thing they did up here. Any question about that part? All right. Viruses make mistakes. And one of the reasons why they make mistake every once in a while is they're not terribly worried because they make a lot of offspring. You don't need every offspring to reproduce to be successful for a virus. And as you can see, the viral DNA that's replicated is a red purple, and then the cut DNA of the chromosome is a purple. If you notice, these viruses get the viral DNA inserted into them, but that virus and that virus, they don't have viral DNA in them. What they have is the purple viral DNA, not viral, a host chromosome DNA of this cell. These cells will replicate normally, will infect a cell normally, and then go through this procedure. But what happens when this cell infects a cell, this virus? it will inject its DNA into the cell, but this isn't viral DNA. It is DNA of this chromosome. And then that DNA can become incorporated into the chromosome of this cell and then expressed. And now the cell has gained new DNA. It's gained genes from this cell. What would happen if this was a cell that had antibiotic resistance, which was on this part of the chromosome, and then that chromosome got put into this virus, and then this virus injected that host part of the chromosome into this cell, and then that chromosome part from this cell got incorporated into the chromosome here, and now it will be expressed. This cell then gains a new gene, which in this case is antibiotic resistance. Any question about any of that? So I mentioned that antibiotic resistance can happen by transduction. It can happen by conjugation. I didn't mention it, but let's say the F plasmid has an antibiotic resistance gene on it. This cell, which was sensitive to it, now gets this plasmid so that both cells are going to be resistant to the antibiotic. And I already mentioned it here. If this DNA, the lowercase a, codes for antibiotic resistance, then this cell's going to become antibiotic resistant when it was sensitive previously. Any question about any of that? How important are those in, well, I guess to a cell or in evolution and mutation of a cell? Spontaneous mutation, I'm just going to abbreviate that. Happens one time, and I'm going to have a hard time doing this. 10 to the 6. Let's go there. Go back. And blow that up. Well, let's go a little bigger even. So spontaneous mutation, you get one nucleotide changing in 10 to the 6 genes. Okay? I guess I don't want that, but I want that part. Come on. For transformation, the occurrence equals one in 10 to the 5th. And in conjugation, occurrence equals, I got to change this. Let's get 2 of them there. So in conjugation, conjugation. It also equals 1 times 10 to the 5th. And then in transduction, it also equals about 1 times 10 to the 5th. So transformation, conjugation, and transduction are going to happen about 10 times more often than spontaneous mutation of this gene. And transformation and conjugation is one way a pre-existing gene can spread in the population. And now, instead of having one cell, which is mutant, we now have 2 cells that are mutant. Any question about any of that? Yeah, let's close that. All right, I did briefly mention plasmids. You should know a plasmid is a small circular piece of DNA outside the chromosome, meaning it's extra chromosomal DNA. It can't contain genes. And here's a plasmid here, but it only has a few genes on it. 1, 2, maybe 3, 4, 5 genes on it. The point is there's only a few genes on a plasmid, and that differs from the genes on a chromosome where we have many, many genes on a chromosome. In bacteria, we have something like 20,000 genes. Maybe that's a human. I don't remember. But the point is, a plasmid is very small compared to the chromosome, and it has very few genes compared to the genes on the chromosome. Plasmids are important because they can replicate independently of a cell, and then the plasmid is more likely to be exchanged between cells, like in the conjugation, as well as in transformation. You're more likely to get a plasmid undergoing transformation or conjugation than you are the entire chromosome. And I guess what's important about plasmids is that they can move around, and this we're calling a transposon. It's a piece of DNA that moves from one region of the DNA to another region of the DNA. A transposon always contains an inverted repeat, and the transposase enzyme, which actually cuts the transposon out and then seals the DNA back, and then cuts a place in the DNA for the transposon to insert, and then the transposase gene anneals the DNA at the cut site, inserting the transposon. So a transposon always has these inverted repeats, and the transposase gene. Sometimes complex transposons can carry other genes with them. Like this kanamycin resistant gene on the transposon TN5, you'll note that there's a kanamycin gene there, resistance gene, and then it has a transposon over here and a transposon over there, and that's usually how we see complex transposons, meaning they don't just have the inverted repeat and then the transposase gene. They actually have the inverted repeat there, the transposase gene there, and then the inverted repeat, and then a complete replica over here. And this can allow the transposon to move from one region of the DNA to another, including moving from the chromosome to the plasmid. And then, as I mentioned, plasmids are important because they allow for the exchange of DNA between cells easier than the exchange of DNA between chromosomes of this cell and that cell. It's much more likely a plasmid's going to exchange. So both transposons and plasmids sort of move around the DNA and then allow it to be shuffled to another cell. So it just speeds up the shuffling of DNA in a population of cells. Any question about any of that? All right, I think I'm going to stop right here. No, actually, we don't have a lab today. Let me go and finish this last slide. Let's talk a little bit about genes and evolution. Genetic variation is differences between genes between individuals, and genetic variation is extremely important to populations. The only way a population can get a brand new gene or genetic variation is by mutation. As I stated, in the human population 10,000 years ago, we had a spontaneous mutation in eye color, and now humans have two different basic eye colors, blue and brown. Maybe someday in the future, we'll have humans with yellow eyes. I mean, dogs have both blue and brown and yellow. Maybe in the future, humans will also have yellow. I don't know. But the point is, genetic variation occurs, and the humans got a new gene, and the only way they can get it is by a new gene, is by spontaneous mutation. But once the population has that existing gene in the population, it can spread around by crossing over, and that just shuffles the DNA, making new combinations possible. But it can move between organisms by transformation, conjugation, and transduction. And then transposition kind of just speeds it up. You transpose a gene from the chromosome to a plasmid, and the plasmid is more likely to go through transformation, conjugation, and transduction. That allows the organisms in a population to now get a new gene. And genetic variation arising from all of these forces drives evolution primarily via natural selection. Any questions about genes and evolution? All right, if not, I'm going to leave it here. And if you have a question, you can see me in the lab from 6:30 to 6:45. Any questions? All right, I'll see you.